co-immunoprecipitation and immunoblotting. So, what is immunoprecipitation, also referred to as IP? Now, this is a technique involving a precipitating protein antigen out of a solution using an antibody that specifically binds to the particular protein. This process can be used to then isolate and concentrate a particular protein from a sample containing many thousands of different types of proteins. Immunoprecipitation, involving the interaction of protein complexes, is then referred to as co-immunoprecipitation, also known as co-IP. Now, co-IP works by selecting types of antibodies that then target a known protein that is then known to be a member of a larger complex of proteins. This targeted proteins, no member is known within an antibody to become possible to pull the entire protein complex out of a solution and thereby identify the unknown members of their complex. This works with the protein involved in the complex bind to each other very tightly, making it possible to then pull multiple other types of members of the complex out of a solution. This then latches onto one member within an antibody. So leading on to what immunoblotting is, also referred to as a Western blotting. This is a type of technique that is an analysis of individual proteins in a protein mixture. Now, in immunoblotting, the protein mixture is applied to a gel electrophoresis and a carrier matrix. That carrier matrix could be something such as SDS page to sort the types of protein sizes, charges, and other types of differences within that type of individual protein bands. The separated protein bands will then be transferred to a carrier membrane. This process is then called blotting. The protein will then become adhered to a membrane, and in that pattern, there has to be a separation due to the interaction of charges. Now, we know that the proteins of the immunoblotting are then becoming accessible to an antibody binding for detection. And so the antibodies within the, for instance, are radioactive labels of an enzyme. This leads to the color types of recognition or immersion of light, which enables the detection of these types of proteins. What are the principal application techniques for co-immunoprecipitation and involves the analyzing of the function and interaction of multiple proteins? It starts off with your protein binding to an antibody. This occurs when you will involve cell lysing starting off. This allows for that to occur through opening and breaking apart the cells to allow the protein to become accessible within the antibody. So now that the antibody is now accessible due to lysing, you then add your antibody to the protein. And once that happens, the added protein A beads are then added to allow there to have pull down upon the antibodies to be pulled and the protein complexes to be separated. When this happens to occur, you want to involve the incubation step. Once the incubation step occurs, you then want to collect it, and once collection happens, you want to make sure you wash the beads, and then you're going to elute the proteins through SDS page loading buffers. This reduces the nature of proteins that have been created within the mixture, which then allows there to be detection of the proteins to be seen visibly. Once this happens, you can then analyze your proteins using SDS page. Improved dilution conditions for native co-immunoprecipitation is the article I chose, and the key question in the article is determining if an elution protocol will allow effective recovery of immunoprecipitated antigen and binding partners, yet avoid elution from the bulk of immunoglobulin. And this experimental design allows the assessment of performance of soft elution protocols using immunoprecipitation of adapted protein complex 1, also referred to as AP1, and the associated proteins. And the left side shows how this works overall during the experiment. And so lane 1, you have the standard elution. Lane 2, you have the soft elution, and the standard elution after soft elution, which is step 3. We're trying to see if there is a change in the elution after soft elution, and if there is, that is a good outlook. And we have our 3 types of adaptive protein complex 1s, which is AP1 beta, AP1 gamma, and AP1 with resulting positive outlooks coming from AP1 gamma. The strengths and drawbacks of this experiment shows how findings suggest that an elution protocol can in fact enhance the co-immunoprecipitation, and this leads to the future research studies should be focused more on the role of AP1 gamma interaction complexes. My question is, what are some similarities co-immunoprecipitation and immunoblotting have? References: